Thank you for the invitation, and good to be among good company. So thank you for coming and listening to that. I'd like to show some work on statistics with counting processes. This is mostly work by a couple of graduate students, Anne-Marie and Gina. Why do we need to work with counting processes? I'll try to convince you that it's a good idea. So let's Washington State University, and Vancouver is not in British Columbia, it's in the state of Washington. So that's where I am. There is another Vancouver too. We're the first Vancouver. Sonia actually already gave a very nice presentation on the spikes and spiking data, and also introduced how several of the difficulties to working with spikes. This is data that's not quite forgiving. It's many difficulties. She spent a long time working with those. We were looking for alternative representations, and in math we like bijection. So if something's exact representation, but may have some nicer properties, we may want to use that. And I want to claim that the counting process. So again, spikes often are modeled as point processes in as a mathematical tool that includes some idea of the randomness. Counting processes, they're essentially the integral of a point process. And so every time you have a spike, you see, okay, I can use that. Every time you have a spike, you raise the count by 1. In the meantime, it continues at the same. So that changes the point events that in the point process to piecewise continuous functions. So they're smoother than before. Once we have piecewise continuous functions, we actually have a lot of continuous time information, and some more standard statistics can be used to analyze the data. Let's see what those might be. Again, an example of a raster representation, multiple neurons firing. This has multiple trials as well. Each bar here is a single spike from some neurons. Already in our community, in the data analysis side, short-term counting processes are used because people do some binning, say 5, 10, 15 milliseconds, depending on the task, and count the spikes in that in a somewhat artificial way. You'll see that the counting processes, if taken seriously, allows for more refined choices in timescales. Here are representations. Again, just looking at the counting process. One neuron, and you look at different samples or different realizations of that process. You get different trajectories. I want to stress again, you get the continuous time, which is at any time that you pick, doesn't have to be binned. You have a value for the count, you have a distribution for the counts, right? And the one-to-one correspondence, when I differentiate that, I get back the spikes, if I need them. That actually allows me for a very nice representation of multiple neurons, because now we have here, I have a number at every time. I can put numbers in lists of numbers for different neurons, and then have a vector of counts that are evolving in time. Here is a representation of 2 neurons as they evolve in time. This is essentially a random walk in 2D. For those specific models here, but anytime you have a spike in neuron 1, you go in this direction. Every time you have a spike in neuron 2, you go in this direction. The trajectory just meanders around. You take a slice. You can look at the spikes and not the spikes, but the counts and the distribution of counts. One of the benefits from counting processes, again, pros and cons. When Sonia said there are the 2 to the n, with n neurons, there are 2 to the n possibilities, that's difficult to process, actually, but provides for a lot of freedom. Here, my complexity grows as n, just as a vector space. So I have, I can only plot 2 neurons in time, but you can easily see how that they can go 100, 1,000, million neurons, whatever you want. On a laptop, I can also easily process statistics for tens of thousands of neurons as they evolve in time. On a laptop, larger things may be bigger computer. So one of, back here, it's easier to see one of, there are some issues with the counting processes. One of this is the count continues increasing in time, right? So that it's always an increasing function. And let's see that here. The other is, as you can also see, the variance may change in time. That depends on the process. Those here are samples from Poisson process. So in that case, we know the variance is proportional to the mean. As the mean increases, the variance increases as well. For networks, that's not necessarily clear, depending on what the network is doing. If it's kind of the dynamics is around a fixed point, talking about stochastic network, the variance is typically limited. So yeah, the mean continues growing in time as the count, but the variance stays in some range. But those, in case they're not, okay, so I should have said that here, fine. I should know my presentation better. Counts increase, variance increases, we can deal with that. Standard statistical tools, what can we do with means? Well, we can center, subtract the mean, and then you have some process in which the mean is gone, and we don't have to worry about a coordinate system that keeps increasing in time. That may still have variance. That's for zero mean processes. Nice statistical word, heteroscedastic, so means variance that changes. But if we do have good model and know how the variance changes, we can use the other statistical piece, which is scaled by the variance, and then we have standardization. Subtract the mean, scale by the variance, we have zero mean, variance one process. It's not counting anymore. Again, there's some very, you see kind of decay in time, jump as a spike, decay in time, jump as a spike. That's subtracting the mean, scaling by the variance is leading to something else. But with that, you see, again, a lot of trajectories that different realizations of the same network or the same process. In this case, we can do statistics in them that's continuous in time. You can take slice. In that case, at least some of the statistics is controlled, and we expect some state, some compactness in state, right? It's not increasing forever. Thank you. Actually, that's one slide from now, but here is an example of the same thing and applying this to network simulations now, not to Poisson processes. So we can, this is simulation. I have more details on the simulation, but network, exciter inhibitory network, it's not Pothian-Disman, it's the Brunel network because it's simpler. Exciter inhibitory, we can run that on exciter neurons, run that on inhibitory neurons, get the same kind of process. What if the rate changes? So that we start going through models now, and so we can have some hypothesis that, well, this may be a homogeneous Poisson process. The rate, the rate of the point process is constant in time. The rate of the counting process increases as a line, right? So it's the rate times time. A variance is equal to the rate in both cases, and we can standardize with that. We can say, okay, maybe it's an inhomogeneous point process, in which case we have to estimate the rate for each time frame, for each time that we use it. That would require replicates to estimate it. In the homogeneous one, a lot less. You just need the final counts to estimate the rates. So we tend to have a lot of data there. But we still have simplification where the variance is equal to the rate, and when we scale by that. Or finally, for individual neurons, again, those are only individual neuron models, we can have non-Poisson assumption, which here it translates just, we estimate the rate, we estimate the variance, and standardize by those estimated values as they are. Again, those interplays, some of them have different data requirements, like this is the easiest to estimate, this requires more data, this requires more data because we need to estimate both mean and variance. They're also good to when you make some assumption, then you look at the residual. One, you can test whether your data satisfies this assumption or not. But here, and for example, you assume this is homogeneous Poisson, subtract the line. Variations along that line may be informative. And so if I do spectral analysis of the residual mean compared to Poisson one, to hypothesis one, I can get some frequency range and try to claim that this is synchronization in parts of the synchronization for neurons, but also synchronization in parts of the network. Here, that's subtracting. That's assuming that the rate, even though it's variable, doesn't make any difference. So I'm subtracting it. I can test hypothesis versus the variance. Assumption here is variance is proportional to the rate, so I can detect whether the actual variance in the data is has this property or not. So the test for Poissonness. This other one, we'll see some results for it. Mostly we work with simulations for now to validate the methods, to test them. At some point, we'll probably develop some more theory or find the papers in the 60s that developed that theory. We'll see. We haven't found it yet. But simulations, working with Marcus's system, NEST, NumPy and SciPy, with all that, Poisson generator to test when things are simulated under our hypothesis, we should get the right results with stuff like that. Integrate and find neurons in the exciter inhibitory network. And the spike recorder is taking subsets usually of usually all the Poissons, but subsets of the network and recording them so we can do additional analysis. Part of the pipeline here is we record spike events, and then there is another part of the code that reads the spike events and does analysis. So if those spike events come from a biological system, the rest of the system is completely agnostic where it comes from. So it's getting ready for data processing as well. The network that we're simulating, on which we're going to do some of the statistics as well, that's, I have the citation here, excitatory inhibitory network proposed by Brunel in the 2000s, used for things like that. Initial idea was to do it as a simple piece of cortical tissue. It turns out to be a little bit too simple for that, but it has some very nice properties, and Brunel and his group did a lot of analysis on it. So we know a lot of results for the network, where again, to use for calibration, to use for validation, it's good to know what we're looking for before we go to data where we don't know what we're looking for, but now we'll have a tool which we trust more. So this is the typical think about 10,000 neurons, parameters as standard. This actually did come from Statistics from one of the examples in the NEST software package. We've tried a few. There is a G parameter here that controls the relative strength of inhibitory to excitatory weights. And so that's the only thing that we change. Once we generate the network, it does have random connectivity, but once we generate it, we freeze it, so the network is fixed. The main source of noise are the Poisson inputs inside the network that we let the Poisson generator pick different implementations of the Poisson inputs. So that's the reason that we have different trajectories in the network, even though everything else is kept the same. Let's see. Right, so some results on the individual neuron level. This is coming from the Poisson generator. So we could have hypothesis one, hypothesis two. We could assume it is actually what we assume and what's happening here. We assume homogeneous Poisson. We're actually simulating homogeneous Poisson. So that should be a good model. But because of noise, when you assume homogeneous Poisson and estimate the mean of the residual, there is a little bit of mismatch, right? We can test this is within thereabouts. I'm just showing what a residual mean would look under an assumption that may be incorrect. If you subtract the actual mean, then of course the residual mean is 0. That's the red line. Right? Same for the covariance, variance in this case. We standardize by the variance. Variance should be 1. We're plotting the estimated variance. In both cases, it is about 1. I'm not sure where the blue thing comes around, but the variance is about 1. So far, those things are working. Results from network simulations, immediately we see that the network is not Poisson. It starts with, the means are fine, we just subtract the means, although there's some places here where the constant rate assumption may be broken. We need to do a more precise statistical estimate. But the variance immediately tells like, okay, we start at 1, but then it converges to about a half. So the variance is half what the Poisson model asserts the variance should be. We scaled by the Poisson assumption, and we get estimated variance of the residual mismatch compared to what the expectation of the model is. So we learned something new about those simulations and about the network. Again, that may not be new to the network. All that statistics can be done with point processes, but the mathematics tend to be a little bit more involved, and computations, at least for now, are a little bit more involved compared to just doing variance per time. So that's on the individual neuron level. Poisson neuron in the network with G equal to 4, that's the standard balanced dynamics of the network. We also tested, tried to see what's the statistics of the residual actually. Because we do things with the process, and now it's not necessarily a known process. So that's where we need to do some theory. So we did some statistics of the residual for the Poisson process, quantile plots. The straight line is where a normal model. Testing now if the standardized model has anything in common with the Gaussian zero mean standard one. And it looks like mostly it behaves like that portion here. In the quantile plot, telling us that it's more variable at the edges than Gaussian, but you look at histograms and kind of expectations under the normal model, we can kind of treat it as the residuals, we can kind of treat as Gaussian models. For the networks, we're seeing, again, immediately, they're actually more Gaussian, that straight part, but they're not the correct Gaussians as we know. There's still zero mean here. The standard deviation is half of what it's supposed to be. Right? So Poisson, the residual after the Poisson assumption is that red line. The actual histogram is a lot narrower than what it should be. But if we scale by, if we do say inhomogeneous, so if we do hypothesis 3, which is estimate the correct variance, then that tells us the residual has a relatively decent association with a normal model. So, yeah.
Just repeat for the upper right plot what the histogram,
here?
What quantity the histogram is?
This is the residual after standardization. So we subtract the mean of the counting process, we scale by the variance of the counting process, and then we see, okay, whatever that is, if we do kind of, we get all the samples for all the times, does it look like a Gaussian histogram of the residual? And also as well, where here we're dividing by the variance of a presumed Poisson process, and it's telling us that's the wrong model. right? Because the variance is lower, as we saw here. So those are a few things that we can do on individual neurons. Okay, I guess that's it. But now it's more interesting to see what is the association between different neurons and network style connectivity. So if all those neurons are Poisson, then the covariance, I'll talk about covariance matrix, that will be the identity matrix in n dimensions, right? So we of this residual, they have variance 1, we standardize to the right variance, they do not correlate in any way, so the covariance of the diagonal elements are 0. We are interested to see what are those covariance matrices for the networks, because I mean, if they're Poisson, great, but that's not very interesting. Hopefully, they're a little bit more interesting. So here are some network covariance matrices. We treat time now as IID. So we're saying we're not going to care that there is some dynamics in time. We just take all the samples for specific time slots, but treat them as one sample. So all the vectors that we get, treat them as one sample for this process. And then all the vectors, you do covariance. We get some covariance matrices. They look like they can come from Poisson, except as expected, the variance is, for this case, is a lot lower. So this should be 1 for Poisson process. In the network, that's the G equal to 5 parameter for the Brunel network. That's running with estimates of the variance a lot lower than what Poisson predicts. Compared to G equal to 8, in which case, again, we get pretty similar structure, kind of diagonal, right? Yeah, mostly identity matrix. If we do statistical tests, they are about the same values, but the values on the diagonal are close to 1.5 rather than 1. So we learned some things about those networks in different regimes. Again, not that we didn't know them, but we can learn them relatively quickly with this approach. That when we first started doing that, that kind of almost diagonal structure was a bit surprising. I was hoping to see a bit more structure in the network. We didn't see it initially. So we progressed, we kind of continued forward. The nice part about that is we now have time resolved, we can do time resolved statistics. So I don't have to scrunch everything and pretend time didn't exist. I can do those slices at any single time that you like. Because I have replicates, I can do covariance. That is more data hungry now. I need enough data to do the covariance for every single time slice. And part of the process here is to try to do some analysis for networks and models in neural systems to identify groups of neurons, timescales on which we can cluster and reuse the data to increase the power of the statistics. In simulation, just one last thing. In simulation, that's not a problem. I just run another 1,000 replicates, I get my statistics. In data, that's a problem.
Quick clarification. How is G calculated? It's some kind of ratio between expectation.
G is selected. So G is the ratio of inhibitory synaptic strength to excitatory synaptic strength. In that network, the synaptic strength is constant. For all the neurons, it just the excitatory strength is some value, and the inhibitory strength is G times stronger.
But it also includes the innervation, so something like the in-degree original?
The original has random connectivity. We could use fixed in-degree or fixed out-degree as well. I'm still using the original network where you just do random auto connectivity, or potentially auto connectivity, but random subset of 10% of the neurons are connected.
Inhibitory weights are 5 times larger.
Yes, yes. Mm-hmm. And the 10% is fixed. And for those simulations, once we generate the weights, we actually keep them. The only thing that we change are different realizations of the Poisson inputs. Poissons are the same rates, but they are random. So when you run the network next time, it's a different set of input spikes. I mentioned that because we will discuss a little bit of that as well. So time resolved. We can produce a movie. In here, we sample every 10 milliseconds, but you can sample anytime you like. So here's a movie of the covariance. I don't know, not very exciting. Something. To remind you, we're roughly here. This is G equal to 5. This is the G equal to 8 in the bifurcation diagram, different dynamic state in that network. So far as Brunel is concerned, we are roughly at the same state of the network, right? We'll see that with this analysis, we see slightly different behaviors. So go ahead.
Asynchronous irregular could have different initial data in what you are calculating to make this emphasize it. Or where are you heading at? What do you want to extract?
Let's see. Again, this is very preliminary results. I was not sure what I wanted to extract, but I knew what I could do with matrices and linear algebra. So I am going to do some of that. Time resolved covariance. We have a covariance matrix. We can do spectral analysis. We can take eigenvalues, eigenvectors, and those we can see the dynamics of those for some of them. So here is again standardization. Here I have couple standardization against inhomogeneous Poisson, assuming inhomogeneous Poisson. Yeah, so I'm subtracting the mean, even if the rate has some information, but subtracting the correct mean, so the residual has really zero mean. But I'm assuming the variance is square root of the mean, whatever that is. Look at the residual variance. Interesting now, previously in the time unresolved case, we were seeing just low variance. Now we see that there was some dynamics, high variance that then quickly entered to low. That might be some initial transients that we weren't exactly careful with the dynamics. This is already starting at 200 milliseconds after the network starts, so initial conditions should not be that much represented, but there is still something there. Eigenvectors are more difficult. This is just one of the eigenvectors. All of those here, 50 neurons, and how that evolves in time. So far as I'm concerned, it's not evolving much in time, even for Poisson processes. When you compute the covariance matrix, when you estimate the covariance matrix, you have some noise. So for the Poisson process, covariance matrix is identity. So anything is an eigenvector. And when you estimate them, some random things may turn out to be an eigenvector. This is what it looks like. And the changes in time may also be slight glitches in estimation. If we standardize by its own variance, we actually estimate the variance and standardize it, we kind of see that reflected. Eigenvalues, they're noisy, but they roughly stay at the same level, not much change in time. After hearing Sonia's talk, I may be more interested to see that may be some signature of synchronization, but maybe not. We can talk more about that. And that, again, is reflected. The eigenvector didn't change much. Again, each one of those is an eigenvector estimated at different time point. Right? If it's noisy, something like that happen. That level of statistics, if you kind of squint, you can say, well, maybe there's a few neurons that are more heavily represented, so more weight at this time period. There may be another few neurons in this time period, but maybe we should just do better statistics. Right?
How do you use the NPI effect?
This is the leading eigenvector. So This is one eigenvector.
Yeah, one eigenvector, and then over time.
Yeah.
But like, what is one eigenvector? Like, I guess you have, let's say, n for an invariant matrix.
I have n by n matrix. Each label is a neuron. So one eigenvector is a linear combination of the counts, well, standardized counts for those neurons.
Yeah. And this, like now you have two matrices, right? So for 0 to 10 and for 10 to 20 milliseconds. Like calculate.
It just add 10 and add 20. I can average some more, but yeah. Yeah.
So now you can.
Yeah.
So now the one corresponding to the largest.
Yeah.
Okay.
The largest eigenvalue.
And then you hope that the eigenvalues are like, so because then you have the dynamics of the eigenvalues.
If you have dynamics, that's interesting. For now, we're not seeing dynamics in the eigenvalues.
Now I can imagine a scenario where an eigenvalue starts up here and goes down, right? Another eigenvalue.
Let me switch to the next slide. You guys are forecasting what I'm going to say. That's good. You have experience. So this is G equal to 8. Similar things. We standardize by assuming homogeneous Poisson, make sure the mean is actually 0, but scale by square root of the mean rather than its own variance. We have something similar, high variance drops down, but then you do get dynamics of the eigenvalues, right? So the eigenvalues have something. I don't know what that means, but they're changing in time. They're changing in hopefully non-trivial ways. What's interesting is that we start getting some structure in the eigenvector as well. This time I don't have to squint too hard. So again, initially you think what you saw before, some kind of, I can call that random. Random loading of neurons to the eigenvector. But here I get essentially persistent loading of this neuron, this neuron, this neuron, this neuron, this neuron, right? And half the time this, we'll see about that. Just stay there. I didn't do anything. This is telling me, trying to tell me something about the network. I'm not sure yet what, but to your question, what if there is dynamics of the eigenvalues? In this case, this is also reflected in a relatively stationary eigenvector. That's also an interesting point. We have eigenvectors, we have projections of the eigenvectors, which are coefficients. The coefficients change in time, but the eigenvectors also change in time. So if you start modeling that and you want to write a differential equation for that, the rate of change of the state would depend on both the coefficient rate of change and the rate of change of the eigenvectors. The coordinate system is turning around to reflect the structure of the covariance. We get to somewhere here and now have a switch, different set of neurons are loading in the leading eigenvector. Now I have a little bit of a story here, a little bit of hypothesis. I'm guessing the leading eigenvalue here switches to the second eigenvalue, right? Possibly. So something like a bifurcation, it's not a state bifurcation, it's kind of variability bifurcation. But I'm guessing this is the leading eigenvector for this guy, and this is the eigenvector for this guy that now became the leading eigenvector. What else I'd like to say? Somehow the network is trying to tell me time frames and time scales. Again, I don't understand yet what they mean and what they're trying to tell me, but I can observe that a time scale of about 200 milliseconds here, I don't know where it emerged from. The time scales in the neurons are 10, 20 milliseconds. This is emerging on a much larger time scale, and the switching is on an even slower time scale. Two things. So again, the first three eigenvalues are interested in that direction. This one possibly switching here. Those two possibly switching here. And I can roughly see that. I should have plotted it, but I guess run out of time. Roughly can see that in the second eigenvector, where you see some of those things, like a roughly twice as fast switching time, where some kind of columns like that appear more often. That has to be taken with a little bit of grain of salt. That's why I'm showing the leading eigenvector. That's unconstrained. Anything else has to be orthogonal to it. And so some of the structure appearing in later eigenvectors is affected by the previous one. The better state, which we're still working on, is to start looking at the low-dimensional projection space, which is hopefully stable. And we'll see what happens when we project the network in, this is a subset of the network that's 50 neurons out of the 10,000, when we project it on a 2-3-dimensional space and see what we get there. Similar things when we actually scale by its own variance. Again, not exactly identical, but still dynamics in the leading eigenvalue, still possible switching between eigenvalues here and there that we need to investigate some more. This is where we take steps every 10 milliseconds, but this is where maybe I should take finer steps to see if something useful emerges. Back to doing the statistics again. When I have something like that, like eigenvector loading, it's fine, getting excited. Eigenvector loading with several neurons, that's an indication that maybe I can combine the statistics for those neurons and have effectively a larger data set. So treat the neurons as IID rather than completely independent things. That's one of those, but if I usually think if I can take the covariance matrix and break it into block diagonal case, then the blocks would allow me to pick up neurons which can be treated as essentially replaceable units, right? And other blocks look at, so instead of one neuron, I have 5 replicates just from neuronal point of view. You see why I may need that. Just so those are the eigenvector is a linear combination of activities of neurons. So that means it has higher weighting in representing this eigenvector. It's kind of more important. If I have if the eigenvector aligns with just one of those neurons, it would just bright yellow here and blue everywhere else. This is showing me that here there are 1, 2, 3, 4. There is some the eigenvectors are normalized, so that there is some scale to the coefficients there. So if I have more neurons participating in the eigenvector, then all those coefficients are going to be smaller.
To up and down to the neurons, it's like they're increasing from green to it's nice in a V node. You put a action passes a node and then it's distributed to different nerves, and the double conductive then increases, but teaches artists.
I can't speculate on that. Those are all point neurons. They like a lot of the structure. Okay. Again, I just don't know.
Are you calculating the covariance over the counting processes or over the residuals?
The residuals. Yeah. Counting processes are unpleasant because the covariance always changes most of the time. We know that. But the residuals, the individual neurons are standardized, their relations are not touched. Yes. So this allows us to do additional standardization. So I can remove that as well.
How different?
This quantifies how different they are from independent. Because independent ones wouldn't have any structure in those vectors from the independent.
It means that those first relation, those, let's say, 4 neurons are brighter, those
They are a lot
are
more correlated.
simultaneously different from Poisson.
Yeah, they are a lot more correlated than Poisson neurons with the same parameters.
This is still for Brunel.
This is still Brunel. Yeah. 2 regimes, we have a lot of work to test all the other regimes, but those are some initial findings in those 2. No, sorry. Yes, yes.
Asynchronous, irregular, whatever.
Yeah, I've looked at some. I look at some rasters. There's not much I can see in the rasters. I should run them in Elephant and see if you can find something. But just looking at them, I haven't found anything else. But yeah, that's why we do those things, because human eyes notoriously see whatever they want. We can use that for inference as well, model-based inference. To again test whether some input that's coming is different from our assumptions or not, and how quickly we can tell that. Here again, we can use simple things, z-statistics or p-value. One reason we're not using t-statistics, for example, is our assumption that again, we are the ones that need to estimate parameters of what neurons are doing, but the nervous system probably knows its own models, if we think of it this way. It has grown with it for several million years, evolution. So the parameters neural systems know, we need data to estimate them. But let's say the nervous system has this idea of what is the correct process under those parameters, and then it's listening to input and trying to tell, is that the same? Is that what they expect, or is that different? Here are some examples. This is mostly for Poisson neurons. Let me see which way. I think, right, the blue one is the one coming from the standard model. We assume that's the case. And then in red, we perturb now the rates for individual neurons. And this has lower rates, so of course it grows slower. And we're trying to use this model to assess when we are convinced that this is not the right statistics. Right? Some examples here. Again, once we standardize, this is just the counting process. We standardize. This is what the blue standardized model looks like. And here is when you standardize an input according to the wrong assumptions. So we still standardize under the blue assumptions, the blue mean and the blue standard deviation, but now it's a process that has a smaller firing rate. And when you standardize with the wrong assumptions, it relatively quickly gets out of the zero mean variance 1 range. Of course, if the rate is about the same, that takes longer. All that relates to how we process information, how we accumulate evidence. When there is a lot of uncertainty, we tend to take longer time to get that thing before we reach a decision. When it's very different, it's easy to say say at some point this is different and reach a decision. So that also gives you time frames, which are behavioral time frames. Once you reach a decision, we can reset the counting start again. Because this standardization, you're dividing by standard deviation that increases in time. At some point, all the effects coming from other parts of the network just decrease and decrease because you're dividing by a larger and larger number. But that says, once I make a decision, I can reset and start counting again, if that's what the nervous system is doing.
So for the homogeneous corpus, the rate is constant, and for the inhomogeneous one, it changes over time?
Yes.
Okay.
Yes.
And why don't I see the change over time reflected in the red curve?
For now, I'm simulating just a homogeneous point process.
Red is also homogeneous.
Red is also homogeneous, different rate, different constant rate.
why don't the means diverge linearly?
Dividing by standard deviation does different. It's 1/square root of time. I'm scaling both processes. I'm scaling under the same assumption that it's blue mean, blue standard deviation. This is very wrong for this process. And the red mean, if I center, the red mean would go down linearly, yes. But when I divide by the wrong standard deviation, that gives you that kind of curve.
Even if it's wrong, it's constant.
It's not. Neither the mean nor the standard deviation are constant. The mean is proportional to time, and the standard deviation is square root of the mean. But the mean increases for a counting process. Anyway, we'll take a little bit later. Let me go to that's almost the last slide. So we can take the z-statistics. This is the z-statistics for those values. So it's average divided by the square root of the number of samples. And we can see how that z-statistics evolves in terms of the z-statistic of the correct process versus the z-statistics of the lower rate standardized under the blue process. So that at some point becomes very different. This is plus or minus 2 standard deviations according to the original model. We can use go that directly with let me see, with p-values. Before I forget, this is cumulative z-statistic. We kind of start observing, and whenever we make a measurement, we add it to the z-statistic, right? So that's accumulating evidence over time. And this is the point that you accumulate enough evidence, you can tell this is a different process statistically. With p-value, very similar. We're testing the statistical hypothesis. Is what I have observed so far, up to here, is that compatible with my model or not? And when I'm observing data coming from the model, it's kind of meandering around, more or less never reaching significance. And when observing data coming from the different rate model, at some point it reaches a state where it says, with high significance, this is not coming from the model that I expect. And so then I have a decision. This is related to like drift diffusion models of decision making, just making it a little bit more model-based here. Essentially the same thing, but on log scale, because it's nicer to look at p-values on log scales. And one last point to make, that looking at just a single neuron. If I have replicates neurons, which means neurons doing the same observation, the same decision and neurons are IID, so they're independently doing the same decision. Essentially, every additional neuron doubles the amount of evidence that I have. And so I can make those decisions faster. Essentially, square root of the number of neurons faster compared to what I have here, because my scaling is different. That's another reason to say in sensory systems to have multiple neurons that look like they're doing essentially the same thing, but in this framework, they help you make decisions faster because each of them is estimating things independently, and then that evidence can be accumulated as if it's independent samples. So instead of time, you do the standard mammalian trick and you spread in space. I think that's where we are. The things I want to stress, counting processes are equivalent representations of network state. We can go back and forth. In some cases, point processes are going to be more helpful. In some cases, counting processes are going to be more helpful. To me, the vector counting were easier to visualize, easier to analyze with continuous tools compared to the point processes. I can get some network statistics easier, And going that way makes it a lot less sensitive to network size. So about n for covariance, that would be roughly n square. The complexity increases like that compared to what Sonia cited, which is 2 to the n when you discretize, and we're looking at binary representations. Initial applications, detecting non-Poisson unit statistics in network, noting the almost diagonal covariance, but also noting some temporal structure in time-resolved covariance, and just features emerging that we don't know what to do with yet, but they're emerging, this quasi-stationary PC dynamics. What I was expecting to see is like a leading eigenvector and has some interesting trajectory in time. What I saw was leading eigenvector, then it freezes in time for a while, then something else freezes in time for a while. I don't know what to make of it yet. Where we're going, more of the networks to test in different regimes. This is just a synchronous irregular, but again, inspired by Sonia, we should look how synchrony is presented in those statistics. The 2-point correlation function that's in neurons and time. So we're looking some covariance between the activity of neuron I at some time and activity of neuron J at some other time, seeing how much structure we can extract there. It'd be nice if those things are time shift invariant, but we have to find out. Additional spectral analysis, some of those things that we're just mentioning here, this bifurcation in leading eigenvalues, if we want to call it that way, where they're happening, how they're happening, why, and the identifying of the smaller subspaces, see if we can get some invariant subspaces there. And this approach to data agglomeration, if we can identify subsets of the network that act as IAD processes, that allows us to collect statistics for physiological recordings without pushing our physiology colleagues to do like 10,000 experiments. Okay, let's say hundreds. 10,000 I'll be last out of the room, but let's say hundreds. Hundreds is also controversial in some cases. And we're starting to look at applying this to data. Our Institute for Brain Science has humongous data sets, so for these still unknown requirements, data requirements, that would be a good first place to start. Thank you very much. I actually wrote that. There you go.
Synchronous, irregular, and asynchronous variable. It's about the activity across the neurons. So we developed on the basis of originally on the basis of Poisson models, what can you measure if you have correlations in the data? Let's assume you have parallel spike trains and you just want to look at, so to say, the individual bins and, for example, count the number of spikes. And in this model, where we were able to include different orders of correlation, we were able to, by the what we call complexity distribution, which is the count on a bin version, count of spikes per bin, and its distribution we can conclude on the correlation structure in the data. Wouldn't this be much easier?
I don't know, but you repeated bins like 5 times. And I'm saying this doesn't need bins. Maybe it's important, maybe it isn't. But that allows for some choices to be made later, possibly. So to do your test, for example, you need to decide bin size. 1 millisecond, 5 milliseconds, 10 milliseconds. But well, I do counting process, but that does not have bin size because the counting process continues in time.
But we have on the bin version.
Absolutely. And I'm saying we don't have a theory yet. Go ahead.
So I found this like here about like the eigenvector of the residual kind of thing that said like that is like basically a co-activation which is more than expected, right? So this
Yes, yes.
Okay, this is right because like I think what often is properly related is that the activity, like you mentioned, you have a probe or like whatever kind of probe that you call the neurons, and actually some of them are proactive, but it's very difficult to figure out which of them are proactive. Right, because I mean, like,
This, by the way, may go both ways. Some of what you're seeing here is a little bit misleading, but they may be more active or less active together because this is absolute value of the results, so that some may come with negative loading, some may come with positive loading.
yeah, but I think still, like, I think
Yeah,
this is something you
yeah,
might
yeah,
need
yeah,
to, like, so this you can explore.
You could have
later.
negative correlations.
Also, if you want to look at the slide, like, it would be much more reasonable than studying the neural network to study one of these clustered versions.
Sure.
Where you have like upstate and downstate, and also examples and
Look,
lessons,
we-
so you don't even have to do something like that. But
We-
you don't even have to like.
We will be trying a lot of those things. We will be trying a lot of those things. This is preliminary results.
Just maybe like you are my this study of the cluster version. So I think like your method, especially
Okay.
this might fit.
I'll ask one of the grad students to simulate that one.
I'm also interested in this plot here in right. I'm really interested in what this is in the network that makes this condition. In the net network, I have not seen something like this.
Mhm.
Where you have some change that then lasts for some, what is time scale is?
About 200 milliseconds.
Yeah,
That's from 100 to about 350.
So maybe with this tool you extracted something that was not apparent to other measures before.
Possibly. Again, some of those things I'm putting them and then I'm describing them. But something that we need to do is do the projections on those eigenvectors and see what's the dynamics of the coefficient. As I said, it in some cases difficult because if the vectors are constant, then the dynamics of coefficient matter. But here the vectors can change in time as well. But in this case, we're seeing they're relatively stationary. So, okay. That gives us a little bit of a window where we can use the easier tools and just track the projections on this eigenvector. I'm actually more interested in the projection in the eigenspace. That's like 2 or 3 eigenvectors. To see what they're telling us about that portion of the network. Very specific one as well, like about 6, 7 neurons.
Can you try to think what happens if you shift like a little bit for a specific neuron? What will happen to the
We need Sony's machinery for that. I don't want to replicate what she's done. So yes, absolutely. So that's anywhere in the network model as well. So that's a very good question, which is network seems to be doing something. How much can we shift the spikes and destroy that? Right. In which case, there are easier ways to find that out. But in which case, we probably get to eigenvector something like that, where there's not much structure. So no, we haven't done that yet.
Okay, no more questions.